egg, and then that fertilized egg will start to divide immediately. And then it will go through the stage of blastocyst and then gastrula, and then further development into an individual, right? You'll never see a stage that the cell will stop. But this scientist, by taking the blastocyst out and give the correct culture condition, they showed that it is possible for you to keep the cells in the pluripotent stage. It's doable. That's actually, it sounds like a very easy task, but conceptually it is important, because only after they demonstrated that we know that you can isolate pluripotent stem cells and maintain them in pluripotency in culture. Otherwise, you would predict that the cells will just continuously differentiate, just like what happens in vivo, right? In animal, it's going to be a continuous process. It's always developing. So that's one stream of breakthroughs that contribute to Shinya Yamanaka's iPSC technology. And then he attributed his discovery also to the somatic cell cloning and cell fusion stream of breakthroughs shown on the top. So, you know, in 1962, Gurdon established that you can use oocyte. You take the nucleus out of the oocyte and you just use the rest of the cell, that you can reprogram a somatic cell nucleus toward the pluripotency. So that is demonstrated later by many other scientists, including in 1997, Wilmot showed that famous Dolly, the clone sheep. So you can clone, nowadays you can clone many species using that technology. But how that contributed to Yamanaka's work? So let me give you the explanation of several historic events, historic discoveries. First is cell fusion. So in 1985, Helen Blau and co-workers found that you can fuse muscle cells with other cell types and making other cell types to become muscle cells. So, you know, in the original work by Gurdon, what's being shown is basically, essentially, you're fusing a nucleated oocyte with a somatic cell, because oocyte is huge. So the cytoplasm is predominantly from oocyte. So you fuse an oocyte, enucleated oocyte with a somatic cell, you make that somatic cell to become pluripotent stem cell. And now Helen Blau wanted to know if other cell types also have the ability, like can muscle cell convert other cell types toward muscle cells by fusing muscle cell and other cell types. Then this experiment design is very smart. So she took mouse muscle cells, skeletal muscle cells, while the fusing partner is human liver cell. In this case, it's human liver cell and mouse muscle cells. Why he picked human cell, liver cell, instead of a mouse cell is because now you can track the two partners. As shown on this slide, the human nucleus, so apparently you fuse the two cells into one, but the human nucleus is usually more evenly stained with a nucleus dye. But the mouse counterpart is punctate. That means you see this dotty stuff when you compare to the human more evenly staining. You can distinguish these two nuclei after the cells are fused together. And then she used a specific antibody against the human muscle protein to show whether that, that reprogramming worked. Why he used the human specific antibody against a muscle protein, a human muscle protein? That is because you have two fusion partners. One is mouse muscle cell. In mouse muscle cell, will you have human muscle protein? Of course not, because it's a mouse cell, right? So that fusion partner shouldn't express human muscle protein. Well, the other fusion partner is human liver cell. The human liver cell won't express a human muscle protein either. So if you use this antibody, none of those two fusion partners should give rise to a staining, a positive staining. And only when that human cell, human liver cell, is somehow converted to become a muscle cell, now you do the staining, you can detect the human muscle protein there. So on this image, it's shown as red. So basically, it worked. That means if you fuse a muscle cell with a liver cell, somehow the muscle cell converts the liver cell to become a muscle cell. See what? Do you follow me? Because now you see a human muscle protein expressed, but the human liver cell cannot express a human muscle protein. Only if that liver cell is somehow converted to muscle cell, it can express that muscle protein. So she showed this works, just like in Gurdon's experiment. Here she showed that muscle cells somehow possess some power in the cytoplasm or in the nucleus. You can tell by this experiment. It possesses some strong activity there. It can convert a muscle liver cell toward muscle cell fate. It's a famous experiment because that experiment showed for the very first time that in addition to oocyte, some other cells in our body also have that ability. It can convert other cell types toward itself, like muscle. And then she went ahead and did additional experiments, additional cell fusions. But the first binding fusion partner is fixed. It's always muscle cell. But the other fusion partner, she chose fibroblast, keratinocyte, and hepatocyte. So basically, fibroblast will be from the mesoderm. Keratinocyte is from the ectoderm. It's your skin cell, right? It's from the ectoderm. Well, hepatocyte is liver cell. It's from the endoderm. So she selected cells from each germ layer and then demonstrated it all worked. No matter you fuse any of these three cell types to the muscle cell, you can convert these cells to become muscle cell. But she claimed in this paper that fibroblasts give the highest efficiency, conversion or reprogramming efficiency, as shown here by the expression of human muscle marker protein. You see that the fibroblast fusion give you the highest efficiency, while hepatocyte give you the lowest efficiency. So the conclusion here, I think she's trying to draw is because the fibroblast is from mesoderm and muscle is also from mesoderm, so they are maybe more similar to each other than other cell types. Therefore, you have a better reprogramming because originally they're quite similar already, and now you just give some push from the muscle cell, some power in there, unidentified at that time. Now you can convert the fibroblast toward muscle cell much easier. But I say maybe that's not a very good conclusion. Can somebody comment on this? Why I said it's not such a great conclusion there? Anybody want to try? I hear nobody. So basically, when you do an experiment, you have one sample from each germ layer, like fibroblast is the only one cell type you pick from mesoderm, and the keratinocyte is the only one you take from ectoderm and liver cell is the only one that you selected from endoderm. You just cannot conclude like that. Just like in our class, we have taller, shorter, female, male, Hispanic, Asian, from all kinds of different groups. If I just take one of you and then I observe something, can I draw a conclusion? Of course not, right? Because apparently each germ layer have many, many cell types. If you select just one as the representative, it's just not that scientific. But at that time, it is a major breakthrough. We have to respect, the limitation in by, by the stage of science development. But I just want to point out, whenever you read the article, you can be critical. You learned a lot already, not only in this class, but elsewhere as well. So you can always, provide some critiques. That is a learning process as well. So it's a great work. It doesn't mean it is perfect. So Helen Blau showed that, you can fuse muscle cells toward other cell types, and now you can convert cell fate. But muscle cell is not so interesting when you compare it to pluripotent stem cell. So muscle cell is kind of abundant, and they generate themselves very well. You go to the gym, you do exercise, you're regenerating your muscle. You're building more muscle. That's a regeneration process. You damage your muscle, but at the same time, your body is going to rebuild your muscle. The outcome is only stronger and bigger. Okay, so you showed that muscle have that power. In practice, conceptually, that's very important. But in practice, you probably don't have a lot of use of that, cell fusion. If you think that as a technology, it's not of much use, because we usually don't run short of muscle. The muscle can regenerate fine. So other scientists want to follow up that work, and now they want to see if pluripotent stem cell have that power as well. So if you fuse somatic cell to pluripotent stem cell, like embryonic stem cell, can you convert somatic cells to become embryonic stem cells? So now, this is some advancement based on the oocyte reprogram. Now you're not using oocyte. Apparently, that's very powerful. You're using just embryonic stem cell. You wanted to see if embryonic stem cell can reprogram a somatic cell type. And this Japanese group, led by Dr. Tada, and did some spectacular work that I will show you. But in this case, he's not going to fuse human cell and mouse cell, and he's going to fuse mouse and mouse cell, mouse embryonic stem cells and T cells. So why he has chosen T cells? Because T cells, like B cells, is very unique when you compare the DNA or the chromosome with other cell types through our body. The reason is because T cells, when they become mature T cells, they had gone through some recombination in their genome. Like shown here at the very bottom, as you can see shown here at the very bottom, the T cell receptor, or TCR, is encoded by a region here, as shown in this illustration, but they pick a small region for the purpose of cell identity. Why? Because during T cell development, one of this region, as I'm pointing to, it's so-called the D exon. We recombine one of these J exons with just not one. It could be one or two or three or quite a few. So it's a recombination that will go on. So you must have a D part, but then the rest will be any numbers of that second part. the J part. So you will have a recombination. Therefore, in the mature T cells, they're going to have different such combinations. So in each T cell, the combination are going to be unique because just like your B lymphocyte, they recognize antigen. So the antigens are different. You need different T cells. So in their genome, it's already rearranged. So you can track those cells because their genome are different from the embryonic stem cell, and it's different from any other somatic cells. It's unique. So when you fuse two cells, you can track one of them by using PCR to detect that region that you're interested in. So here, it's just to show you the example. Like in T cells, this is a mixture of T cells. So if you amplify this region, in this lane, this T indicated lane, this is a mixture of many T cells. It's just extraction from your circulation. You get T cells, and then you do a PCR of that mixed population of T cells. Of course, you're going to see many band. Why you see so many band is because, well, you have different recombinations in different cells. So that's why you see this mixed discovery. And then if you try to do the same PCR for ES cell, the ES cell won't have any recombination. So basically this region is just like shown like this. So you have exactly a 1.8 kilobase per band if you do a PCR. So you don't see any of the smaller band. But then if you isolate individual T cells, what you're going to see is you have two alleles, right? So in one of the allele, it's going to be properly intact, while in the other allele, you see different band. The different band indicate different recombination in the T cell receptor locus. And now you have one way to track the T cell. And then he used another way to track the T cell as a fusion partner. And he basically put a OCT4 promoter-driven GFP as a reporter into the T cells only. And here, at this point, I hope you guys have already learned that OCT4 GFP is just an indicator, and this is done through maybe a homologous recombination or by other ways of transgenic. You put this gene into the T cell nucleus, okay, into the T cell genome. And now, since it is a T cell, of course, it's not pluripotent. Therefore, in those T cells, you won't see fluorescence. You don't see GFP expression because the OCT4 promoter is not active. It's only active in pluripotent stem cell, not in T cells. So to show that point here, they isolated the animal. They have developed the animal harbors this reporter. But in thymus is where you isolate T cell. It's the organ for T cell. In thymus, you don't see green fluorescence because T cell shouldn't express OCT4 or GFP. But if you isolated the ovary from that animal, you do see that the ovary is lighted up by green fluorescence because in the ovary have a lot of, germ stem cell. In germ stem cell, OCT4 is active. Therefore, in the ovary, you can see green fluorescence. So the top row here, just to demonstrate they did some manipulation in the T cells, and that manipulation serves the purpose. So that means in T cell, you don't see green fluorescence, but if somehow the T cell get converted to become pluripotent stem cell, you can detect fluorescence. However, no manipulation is done in the other fusion partner, that's embryonic stem cells. So the embryonic stem cell don't have any genomic manipulation. So if you see any green fluorescence, it must from the T cell because in other fusion partner, there's no such manipulation. They don't group green fluorescence whatsoever. So that's what the background I explained, why they picked T cell, and then they did further manipulation of the T cell. And now they did the cell fusion. They fused embryonic stem cell and T cells. And then after cell fusion, they put the cells in culture. You see this typical, island of nice embryonic stem cell culture, right? But in some of those nice colonies, you see green fluorescence. What does that mean? That means, okay, the T cells somehow have the OCT4 locus activated. The T cell now have activated OCT4 promoter. Therefore, the GFP is now expressed. That means the T cells get converted. I hope you can appreciate the technical detail. Otherwise, this is too simple. If you just read this like a history book, then we lose all the purpose of me teaching you. So I hope you understand, the technical details here, which is not difficult actually. And then you must understand now you fuse the two cells. Both cells are 2N, right? The embryonic stem cells 2N, well, the T cells also 2N. You fuse them together, you create a 4N cell. The 4N cell apparently is abnormal. It's not normal cell. Normal cell only have 2 copies of chromosomes, one each from each of the parental side. So here they showed the karyotyping. Basically, you have done the cell fusion, and now you isolated the metaphase chromosome and try to check if you have 4N here. The answer is yes, you have 4N, but there are some abnormality. Like there's some extra. No, it's not extra. This 2N somehow is fused to one of the X chromosome. So this happens when you do a cell fusion. Sometimes weird thing happen. In this case, chromosomes get fused together. This is something you can't avoid. In some of the cells, it's going to have abnormality. So this is not ideal for any therapy purpose. It's only conceptually important because you don't want to create any cell that's foreign. That's totally unnatural. It may cause problem, although even if we don't know what kind of problem it may cause, but it will cause problem. But anyway, it doesn't matter. Now you have a 4N cell, it's a cell fusion. The authors here want to demonstrate, okay, you said you have converted the T cell toward the pluripotent stem cell. Can you demonstrate that those T cells, the stem cell originated from the T cells can differentiate toward 3 germ layers? So they did this. They moved ahead and did a chimera experiment. So basically, this hybrid ES cell, it's a 4N cell, ES and T cell hybrid cell. They just do a blastocyst injection anyways. They hope this 4N cell can still somehow, differentiate, develop, and participate in blastocyst formation. And the answer is yes, they did. The cells can actually participate in blastocyst formation. So you have this later embryo as shown here, but this blue color is only another manipulation in T cells. So okay, so this beta-gal gene is put into the T cells. This beta-gal gene is put into some promoter of the T cell, not in embryonic stem cell. So when you see this blue color, that means, well, this T cell can participate. I should say the pluripotent stem cell created from the T cell can actually participate in development. So let's see the details. So when you see this MES, that means it's mesoderm, a cell from mesoderm. When you see ECT, it's cell from ectoderm. The ectoderm in this developmental stage actually is in the internal lining of the embryo, while the endoderm by name, it should be inside, but endoderm at this developmental stage is actually at the outside of the embryo. So they have shown that some of the endoderm cell, some of the ectoderm cell, and some of the mesoderm cell are from the T cell by this technique, because you can track the T cell. So this just demonstrated that by fusing embryonic stem cell and T cell, you can make the T cell to become pluripotent. You know, the evidence here is, okay, the T cell now become a different cell identity, but that cell identity can give rise to 3 germ layers. That's the, you know, one of the criteria of pluripotent stem cell. So this is another very important experiment. So to summarize what I have explained, you know, at one point of you know, in the history of stem cell research, scientists used cell fusion, and then they found they named the fusion partners as responding cell and dominant cell. So the dominant cell will be embryonic stem cell or muscle cell in our 2 examples, while the responding cell will be fibroblast, T cells, B cells, etc., like we have explained. If you fuse these 2 cell types, the cells are going to undergo a process we later call reprogramming. So basically, you have 2 cell types, but one of the cell type will play a dominant role. That means it's going to make that cell indicated as blue to somehow have epigenetic change similar to the fusion partner, as shown here. So, but in this kind of cell fusion, you could have 2 outcomes in terms of the nuclei. So in one situation, that 2 nuclei could fuse to become 1 nucleus. In that case, we call the fused cell a synkaryon, synkaryon, synkaryon. But in other cases, the 2 nuclei are always going to be separated. So they don't fuse toward a single nucleus. Like in this case, we call this heterokaryon. Heterokaryon, that means the 2 nuclei are never going to fuse together. So they are going to be independent, but they are not functionally independent. One of the nuclei is going to be dominant. So the genes encoded from that nucleus is going to somehow reprogram the other nucleus. So the other nucleus will have epigenetic changes. So originally it's a T cell, they express T cell gene. Now they're going to shut down the T cell gene. They're going to activate the pluripotent genes, such as OCT4. Okay, so that's one line of another line of evidence, as I have explained, that's by cell fusion experiment that Shinya Yamanaka said that contributed to his breakthrough later. But in those experiments, it is impossible to know what factor actually contributed to that reprogramming. You know that those dominant cells have power. The power are absolutely there. But what are the factors? Are they proteins or microRNAs? What are those things that reprogrammed the cell toward a different identity? Therefore, you know, in history, some other scientists demonstrated other, you know, very important information that showed that they have identified some factors. Okay, so here I'm going to explain to you the 3rd stream of important events that led to the iPSC technology. So that is done in 1987, one by Goering and the others by Weintraub. I'm going to explain to you the work by Weintraub. And some said that Weintraub should be the one who won Nobel Prize because the work that I'm going to explain to you later. But this scientist unfortunately died at a young age. He was or 49, and he had a brain cancer. I believe it's glioblastoma, like one of the one of the worst cancer that even today we don't have a very good treatment for it. And he was an athlete. He played quarterback for college, competitive football, and he played baseball, and he's a very, very basketball player as well. So he was playing with one of his friends, and he usually made very difficult shot easy, but on that day he couldn't shot some of the easy ones like a layup. He missed quite a few of those, and then his friends felt that something was wrong, and then it took him. They both work in Seattle. There is a famous organization called the Fred Harkinson Cancer Center. I believe it's one of the best cancer centers around the world, and then they work there. So some doctors, they give a checkup. Actually, it was Dr. Tab Scott, who was a postdoc, a previous postdoc at Weintraub. So he checked him up, and he couldn't see from an area of his eye. He partially lose eyesight. It's a sign, of a neurological problem or a tumor in the brain. And then just 2 days later, he was in the operation room, and then one of the best neurosurgeon performed the surgery, but still it was that fast. It's only several months. I felt maybe it was like 5 months or so, and he passed away. And this is one of the most brilliant scientists, and his finding actually was I would say it's the major I would say it's the immediate influence toward Shinya Yamanaka's work. So why? Let me explain to you. So basically, he followed Helen Blau's work. In Helen Blau's work, she fused the muscle cells and other cell types and showed other cell types can be converted toward a muscle fate. So Weintraub want to know what are the factors in muscle cells that are so powerful that can pull other cell types toward a muscle fate. But then he used a different system. What he used is mouse embryonic fibroblast. So you heard about this many, many times. Mouse embryonic fibroblast is a widely used cell type. So how you prepare mouse embryonic fibroblast? Basically, you take an embryo. It's a mouse embryo of about 13 day old. You take the embryo, you just mince the embryo to become a single cell suspension. You put the cell in culture, some cell will attach to the culture surface, and then you call the cell mouse embryonic fibroblast. So you see that the process is quite simple and raw. It's not very selective. So in the preparation of mouse embryonic fibroblast, it is observed that sometimes when you culture the cell to confluence, that means you make the cells to be in contact of each other. You leave no free space in the culture surface. Every cell is in contact with other cells in cell culture, which is called confluence. And now you can observe that some cells become muscles. So if you don't want to make the cells to become confluent, and then you can treat the cell with a chemical called azacitidine. Azacitidine is just a chemical. You treat the cell with azacitidine, you can make the cell to become muscle cell as well. So that's fascinating. So you can make a fibroblast to become muscle cells. It's a kind of if you go through the road of from sparse culture to confluence, then that's a natural occurring process, right? But if you treat the cell with azacitidine, you did manipulation. You change the cell from fibroblast to become muscle cell. And then Weintraub was curious, what's the major molecular mechanism there that so easily make a fibroblast to become muscle cells? So he have a simple idea, but technologically it's very difficult. Nowadays, we can easily compare the gene expression pattern between two cell types by doing sequencing. You just do an RNA sequencing, you get that information immediately. But in the old days, he had to use some other technology. I won't explain to you how he did that, but he was able to identify that there are not many genes that are differently expressed in these two cell populations. One's fibroblast, there is fibroblast derived muscle cell. Okay, so he compared these two populations. He found there are a handful of genes that are differentially expressed, and with MYOD, MYOD was the most abundant one. So MYOD is only expressed in muscle cells, not in fibroblast, in which the muscle cell was originated. So he found, okay, maybe this handful of factors are important factors. Let me try to put MYOD, just one of these factors, into the fibroblast to see what will happen. And he put MYOD by transfection. You put a plasmid encoding MYOD into the 10T½ that's the name of the embryonic fibroblast. Put in there, wonders happened. So he showed that those cells start to express MYOD. Of course, you transfect in MYOD, the cells are going to express MYOD. But in addition to MYOD, other muscle-related genes, such as MYO-A, MYO-H, MHC, the myosin heavy chain, and myosin light chain, MLC1. So all those muscle-specific genes are now get activated by a single transcription factor MYOD. You put MYOD into fibroblast become muscle cells. So that is such an important breakthrough. It demonstrated for the very first time that there are powerful transcription factors in cells that maintain or determine the cell fate. So if you take that powerful factor, you put it into other cell types, there is a possibility that other cells will get converted toward the cell fate. So this is such an important discovery, but unfortunately, this scientist couldn't, further advance his finding because of his illness and death later. But Shinya Yamanaka was great. That's why he won Nobel Prize. The reason is because once the scientists, Tada and Suori, did that, the embryonic stem cell and T cell fusion, and scientists still feel, once they have this Weintraub's experiment, scientists still feel like maybe it's easier to make muscle. Okay, the muscle is much simpler. In cell identity, the muscle is just doing contraction. You need maybe limited amount of proteins to get activated, so the muscle will be able to function. But embryonic stem cell is much more complicated. So we don't know, the function or what genes are important for the maintenance or for the determination of embryonic stem cell. So like I shown on this slide, before 2006, the prevailing will there is nuclear reprogramming to a pluripotent state is a highly complex process that might involve cooperation of up to 100 factors. To identify those 100 factors will not be an easy task. How can you identify, the 100 factors that coordinate to maintain and determine the pluripotent stem cell state? It is hard. Then it comes Shinya Yamanaka. He knows that people thought there must be 100 or more factors that are involved in determining the fate of pluripotent stem cells. But he felt, let me just do something. Let me try this 24 factors. Although there are many factors that are unique in stem cells, but let me try this 24 factors. So he handpicked these 24 factors out of his genius mind, I would say, because there could be many other factors involved as well, but he picked this 24 and he started that experiment that our one of our student-led group, discussed. But here I just want to reiterate that process again. He started with 24 factors. He put into mouse embryonic fibroblast. He found that these 24 factors worked. You can have colonies. The colonies are pluripotent. And then he want to narrow down. And then what he did is 24 factors minus 1. Take one factor out, one at a time, to see which factors are necessary, essential in the process. And then he found that 10 factors are absolutely required. You cannot reprogram cell without the 10 factors. And then he narrowed it down to 10 factors. He started to use 10 factors to do the reprogramming. And then in this process, he found that 10 factors are sufficient as well. It can convert cells. So he started to remove one at a time. And then he found that 4 factors are absolutely required. So then eventually he used these 4 factors, that is SOX2, OCT4, KOF, and CMake, to do the reprogramming. And these 4 factors are called Shen-Yang-Yamanaka factors. So now I hope some students can make a comment if this is a great strategy. If not, why? Is there any other alternative method to design an experiment? Remember, your participation will be graded. I have like 2 to 5 students in my mind who are going to have a good participation score, class participation score. They're going to receive A for that part, but the rest of the group at this point, it's not up to my expectation. You are not going to receive the A for that part. Okay, at the end, it's going to be I put all the score together to determine your final score. So if you can participate, you followed what I said, please participate. Otherwise, don't expect a good score there. I would say this is not the greatest strategy, because when you put 24 factors into the cells, can you track them?
No.
It's very difficult.
No, you can't.
Yeah. So when you say you have 24 factors you put into this cell culture, how many factors are actually into the cell? It's a big question mark there. But because he's successful, and nobody will question that later on, but we can always question. That's you can judge an experiment by your experience and by your knowledge. So for me, I know it is impossible, almost impossible, to put 24 factors simultaneously into cells.
Professor,
Yes.
I believe when you put the 24 factors at a single time, like in one cell, I think it makes the cell confused and to the way it activate. But when you use the 4 factors, it more relevant that the
Yeah, I
cell
think
could
you have
catch it.
very good points there. It could be a possibility, because all the 24 factors have different functions. Although they're all related to pluripotency, but maybe they're related to pluripotency in different ways. Like some are related to cell cycle, some will be related to metabolism and such. So like you said, if you have 24, maybe the cell get confused. It's a possibility there. Okay.
So what would have been more like a more efficient way to do it?
Well, I don't know what is more efficient way. This is quite efficient because he picked the 24 factors, but to start with, why this 24 factors? If you pick like another 24 factors, it could be equally effective or maybe even better. Just that nobody has tried, it doesn't mean theoretically, other factors won't be better.
I see.
Right?
Yeah.
And also, because there are hundreds of ESL specific factors, he picked this out of, as I said, his genius mind. Of course, he was successful. We must give him credit. But theoretically, there may be some other better ways. Like you said, if you think 4 factors are good, why don't you do a random combination 4 factors out of this 24?
Oh, that would have been a lot, though, I think.
That would be a lot, but still, that probably will allow you to come up with a better, higher efficiency reprogramming. And another thing is when you do this 24 minus 1 strategy, actually you're confusing two concepts. One is sufficient, the other is necessary. Right? Those 10 factors after the first round, you can say they're essential factors. Without them, you cannot do reprogramming. That doesn't mean they are the sufficient factors.
That's true. Yeah.
There's a conceptual confusion in there because, like I can give you an example. I give you this lecture. I'm not independent. I'm not essential because I can get replaced by another professor who is in this field, right? I'm not essential, but I would say internet is essential, right? If you don't have internet, nobody can do this, right? You have to rely on internet. But can internet teach this class? Of course not, right? Because internet is an essential factor. It doesn't mean it can do the job by itself. So conceptually, there are something there also. So at the beginning, they get criticized, but we have to say, their discovery is a major breakthrough. There's no doubt about that, but it doesn't mean it is perfect. We could come up with other better combinations if we try very hard, if we have a lot of resources. So this is from the paper again. The scientists used this as your reporter. So they put a beta GEO, like I said, it's a drug resistant gene as well as a beta gal as a reporter. They put into this locus. They were able to select the drug resistant clones. That's what the students have discussed in their discussion. And then in this slide, what they have shown is the cells can differentiate towards 3 germ layers. The cartilage is from mesoderm, CNS, a central neuron system, is from ectoderm, muscle mesoderm again, adipose mesoderm, and epithelium ectoderm. So I criticize this figure because they don't have any representative of endoderm. If they have some gut.
Yeah, actually, when we did the presentation, we looked at it. And we realized that we didn't want to include this picture into the presentation because we didn't find anything on the endoderm, and we were like,
very good. Yeah. If you had that in your mind, it's very good. But then they did that's in vivo, in animal differentiation or development, right? And then they also did the differentiation in vitro. They cultured the cell and induced them into different lineages. And now they have 3 germ layers. They show that the cell can become smooth muscle cell. They can become liver cell. The alpha fetoprotein is a liver cell marker. Okay, now they show that they can become liver cell. And the beta-3 tubulin is neuron marker. That means the cells can differentiate in vitro into neuron lineages. Okay, and then you explained this in your presentation. I just want to point out here. You see the MEF. This is the analysis. The F panel is the analysis of the DNA methylation, as I explained earlier. Each circle is a CPG. If the CPG is methylated, then it's a filled circle. If it is unmethylated, it is an unfilled circle there. You can see in mouse embryonic fibroblast, you see a lot of methylation in the promoter of OCT4, right? In embryonic stem cell, you don't see many methylation there because OCT4 should be very active in embryonic stem cells. But after their reprogramming, they have this iPS cells, 3 different representative of the iPS cells, do you see that the DNA methylation is converted to the ES-like? Not really. The OCT4 promoter remains to be similar to mouse embryonic fibroblast. So although they published the work, they showed that the cell behave like embryonic stem cell, but if you study the molecular detail, the promoter of OCT4, they are not converted. So the conversion is not perfect. And then later on, they used a different selection strategy. And now they put a GFP underneath the promoter of NANOC. NANOC is another pluripotent gene. And then they did the selection this way. So you still pull in the earlier experiment, they did a drug selection. Now they did a GFP selection. You just capture the green fluorescent cell, but also this GFP is under a different pluripotent stem cell gene, okay? And from this selection, they showed actually now the MEF cell, the promoter is methylated for OCT4 and OCT3 pluripotent gene. In embryonic stem cell, you don't see methylation. Now, with this new selection strategy, the resulted cell also have a very good pattern of methylation, just like embryonic stem cell. So the selection strategy matters. But those work were all done in mouse embryonic fibroblast. Now, this group in 2007, that's 2 years later, reported that in human, and this is the same group which established the human embryonic stem cells from Thompson out of University of Wisconsin. Thompson's lab reported that you can use OCT4 SOX2 NANOG and LIN28. So these two are different, although they also have OCT4 and SOX2, but they don't have KOF and CMYC. Instead, they have NANOG and LIN28. They show that these four factors can reprogram human fibroblasts toward pluripotency. And they started with 14 genes instead of 24. They started with 14 genes. So you see this, you have the freedom. You can choose 100 genes, 15 genes, 24 genes. That is not some different scientists could have different opinion of what to choose. So I guess what I'm trying to say is Shinya Yamanaka made a very important contribution to the stem cell field and to biology overall as a whole. But conceptually, it is so important. I cannot overemphasize the importance of it. But it doesn't mean that it's the best reprogramming strategy. You could come up with other factors that may do an even better job, but those will belong to the better strategy, will belong to the future, to us, to people who follow the work to build up more efficient strategies. He was the pioneer. He did the conceptual breakthrough. So here is a comparison of the several reprogramming strategy. You can read through this. Apparently, I don't want you to memorize the technical details, but it's fun to compare them and see which one is more efficient, which lead to what kind of changes in details. So that's the end of today's lecture. Thank you. If you have questions, you can stay behind.
Yes, Professor. I have a one